other lecturers, they have already made time for the lectures. So if we keep pushing the lectures backwards, it might create confusion. So what I will do for now is post recording for gluconeogenesis. It is a very small topic. In fact, just like, yeah, it's a very small topic. Most of the reactions of gluconeogenesis are the reactions. Very small topic. So I will post a recording of that, and then once all the topics have been covered, we can do a makeup lecture for gluconeogenesis. Yes? Okay. So yes, so today is the last topic in carbohydrate metabolism. From next week onwards, you're going to start with lipid metabolism. So here, glycogen metabolism, as the name suggests, glycogen, we all know it is a polysaccharide, isn't it? It's the storage form of glucose. So in animal tissues, excess glucose is stored in the form of glycogen. So today we are going to discuss the synthesis of glucose as well as the degradation of glucose. So the two pathways together make glycogen synthesis. And glycogen, it is a storage form of glucose. It is a polysaccharide. Glycogen metabolism, it occurs both in the liver as well as in the skeletal cells. So the major or the main tissue or main organ in which glycogen metabolism takes place is the liver and the muscle. So these are the two main tissues in which glycogen metabolism takes place. But small amounts of glycogen are synthesized in the brain, heart, kidney, adipose tissue and erythrocytes. But the amount of glycogen that is produced in these tissues are very minute. It's a very small amount. The majority of the metabolism takes place within the liver and muscle. Now, because your muscle mass is greater than that of liver, the amount of glycogen stored in the muscle is greater than that stored in the liver. Because your skeletal muscle has more mass than your liver, isn't it? Liver is just a small organ, but your skeletal muscle, it's present, well, it's made up of, or it covers your entire body. So the muscle mass is greater than the liver, and because of that, the amount of glycogen which is stored in the muscle is greater than those stored in the liver. Now, the glycogen stored in the liver has a different function than the glycogen stored in the skeletal muscle. The importance or the function or the importance of the glycogen that is synthesized and stored in the liver is so that it can provide glucose to maintain normal glucose level. So whenever your blood glucose level drops, the glycogen that is stored in the liver, it will be catabolized, or it will be degraded, or it will be broken down to glucose, which will then be released into the blood so as to maintain the blood glucose level. Clear? The glycogen that is stored in the liver, the function or the purpose of that glycogen is to maintain the blood glucose level in between meals or during fasting period. So when your blood glucose level drops, the glycogen that is stored in the liver, it will be broken down into glucose, which will then be released into circulation to maintain the blood glucose level. So this glucose is carried to the extrahepatic tissues where it can be used as a metabolic fuel. Whereas the glycogen that is stored in the muscle, the function of this glycogen is to provide glucose for glycolysis that can be used by the muscle itself. Clear? So whatever glycogen is stored in the muscle, it is broken down to glucose and it is used by the muscle itself. It is not released into circulation. Yes? So only the glycogen that is stored in the liver will be broken down into glucose, which will be released into circulation. Whereas the glycogen that is stored in the muscle, it will be broken down to glucose, which is utilized by the muscle itself as a metabolic fuel. Clear? Yes? So glycogen metabolism, it takes place within the cytosol of the cell. So just like glycolysis, glycogen metabolism also, both glycogen synthesis as well as glycogen breakdown takes place within the cytosol of the hepatic cells as well as the skeletal muscle cells. So it takes place in the cytoplasm. Now, cytosol. So in the cytosol, the glycogen along with the enzymes that are required for glycogen synthesis and glycogen breakdown, they are all packaged into granules. So within the cytosol, you will find granules which contains glycogen plus the enzymes that are needed for its synthesis as well as breakdown. So together they are packaged into granules which are present within the cytosol. So liver glycogen, the stores increases after a well-fed state. So as soon as you've had a carbohydrate-rich diet, the excess glucose that is present in your body will be synthesized. The excess glucose, it will move to the liver as well as the skeletal muscles, and in the liver and skeletal muscles, this excess glucose, it will be converted to glycogen and it will be stored in these tissues. Clear? So in the liver, the liver glycogen stores, it increases following a well-fed state or after a carbohydrate-rich diet, and it will be depleted during fasting, because when you're fasting, you're not consuming any food, isn't it? Yes? So in order to maintain the blood glucose levels, whatever glycogen is stored in the liver will now be broken down to glucose. So your glycogen reserves or your glycogen stored in the liver will be depleted. Clear? So in a well-fed state, your glycogen reserves in the liver will be will increase, whereas during the fasting state, your liver glycogen reserves will be depleted because it will be broken down to release free glucose into the circulation in order to maintain blood glucose levels. Whereas in the muscle cells, the muscle glycogen is synthesized following a well-fed state. So just like liver glycogen, muscle glycogen is also synthesized following a well-fed state because there is an abundance of glucose. So this excess glucose will be stored as glycogen in the muscle cells. Now the muscle glycogen reserves are depleted following a strenuous physical activity. Because during physical activity, we discussed this in glycolysis. During strenuous or prolonged muscular activity, glucose is constantly required, isn't it? In order to perform glycolysis to give you ATP molecules. Yes? So whatever glycogen is stored in the muscle, when you are performing muscular activity, it will be broken down to glucose, which moves into glycolysis or which is channeled into glycolysis so that ATP molecules can be generated. So following well-fed state, your muscle glycogen reserves increase, but it is depleted following strenuous physical activity. Clear? Whereas the liver glycogen is depleted following or during fasting period. Yes? Hope that's clear. So yeah, so so as I mentioned, glycogen metabolism, it occurs within the cytosol. So why do we prefer the storage of glucose as glycogen? Because excess glucose can also be stored as triacylglycerol. Because excess glucose undergoes glycolysis and the pyruvate is converted to acetyl-CoA. This acetyl-CoA can then move into the synthesis of fatty acids. So it is possible to store excess glucose as triacylglycerol or as fatty acids. But why do we prefer the storage of glucose as glycogen? It has three advantages. So the first is fatty acids cannot be mobilized or released from adipose tissues as quickly as glucose can be released from glycogen. So fatty acid the hydrolysis of triacylglycerol to free fatty acids, it takes longer, or it takes more time than breaking down glycogen to glucose. So by storing glycogen, or by storing glucose as glycogen, it is easier to release glucose during fasting period. Clear? Yes? So fatty acids, it cannot be released from triacylglycerols as rapidly as glucose can be released from glycogen. So that's one of the advantages. Second is fat or fatty acids cannot be oxidized in the absence of oxygen. We know that glycolysis can occur under aerobic as well as anaerobic condition, isn't it? So even if oxygen is not available, your cells can still carry out glycolysis to produce ATP molecules or to produce energy. But in the case of fatty acid oxidation, you do require oxygen. Clear? So that's another advantage of storing glucose as glycogen, in that glucose can be oxidized even in the absence of oxygen to produce ATP molecules. The third one is fats cannot be converted to glucose, and as I already mentioned during glycolysis, glucose is one of the most important metabolic fuels in tissues like the brain, erythrocytes, because they cannot use fatty acids as a metabolic fuel. Clear? So that is another advantage of storing glucose as glycogen, so that there is a constant supply of glucose to the brain as well as erythrocytes, because these cells cannot use fats or fatty acids as a metabolic fuel. Yes? So these are the three advantages of storing glucose, excess glucose as glycogen. Next, we move on to glycogen biosynthesis, which is also known as glycogenesis. So please remember these terms: glycogenesis. Glycogenesis meaning glycogen synthesis. You need to know these terms. So glycogen synthesis in the muscle as well as in the liver, the reactions are the same. So you have the same reactions and same enzymes both in the liver as well as the muscle. So glycogen synthesis, it begins with glycogen. So once glycogen enters into the once glucose enters into the hepatocytes, that's the liver cells or the skeletal muscle cells, glucose will be phosphorylated to glucose 6-phosphate. So this is the first reaction of glycolysis. Hexokinase in the muscle and glucokinase in the liver will phosphorylate glucose to glucose 6-phosphate. So here you need an ATP molecule, because the ATP molecule is going to donate the phosphate group. So here in the liver, hexokinase, and in the skeletal muscle, in the liver, glucokinase, and in the skeletal muscle cells, hexokinase will phosphorylate free glucose into glucose 6-phosphate. Then in presence of the enzyme known as phosphoglucomutase, this enzyme will just transfer the phosphate group, which is attached at C6 position, to C1 position to form glucose 1-phosphate. I hope these steps are clear. Yes? First step is phosphorylation. You need an ATP molecule, because an ATP molecule will donate the phosphate group. The second step or second reaction is the transfer of the phosphate group from C6 position to C1 position. Yes? Now, in order for glucose to participate in glycogen synthesis, it needs to be activated. So, glucose 1-phosphate, it cannot participate in glycogen synthesis. It needs to be activated, and the activated form is known as UDP glucose. So here what pretty much happens is glucose 1-phosphate will condense or will react with a UTP molecule. So UTP is uridine triphosphate. It is one of the nucleotides. So glucose 1-phosphate, it will react with UTP to form UDP glucose in presence of the enzyme UDP glucose pyrophosphorylase. So before we move on, I just wanted to mention, so you have kinase enzyme and phosphorylase enzyme. So these are two classes of enzyme. Whenever an enzyme ends with kinase, it means that it is involved in phosphorylation reaction. Clear? Yes? Phosphorylation reaction is where there's addition of a phosphate group. Yes? From a phosphate group donor like ATP. Yes? So whenever you see the name kinase, like hexokinase, glucokinase, or later on you will find other types of kinase like protein kinase. So whenever you see the name kinase, it means that it is involved in phosphorylation reaction. Clear? The addition of a phosphate group to the substrate. Then the other type of enzyme is known as phosphorylase or phosphatase. So you have kinase. Kinase enzymes are involved in phosphorylation reactions. And then you have phosphatase or phosphorylase. So phosphatase or phosphorylase enzymes, they are involved in dephosphorylation reaction, that is the removal of phosphate groups. Clear? So just remember these two types of enzymes. They will help you know what type of reaction or what the substrate. Whenever the enzyme is termed a kinase, it means it is catalyzing a phosphorylation reaction. That means there is an addition of phosphate to the substrate. Whereas phosphatase or phosphorylase enzyme will remove the phosphate group from a substrate. Clear? So here, UDP glucose pyrophosphorylase. So here it's a pyrophosphorylase, so meaning a pyrophosphate. So pyrophosphate is two phosphate groups. So it is two phosphate groups joined together. So that's known as a pyrophosphate. So here UDP glucose pyrophosphorylase produce the product or produce the intermediate known as UDP glucose. So one phosphate from UTP and one phosphate from and the phosphate from glucose 1-phosphate are removed as a pyrophosphate group to give you UDP glucose. Clear? Yes? The phosphate that is attached to glucose 1-phosphate is removed along with one of the phosphates of UTP, because UTP means uridine triphosphate, isn't it? It has 3 phosphate groups. Yes? Whereas here in the product it is UDP, meaning uridine diphosphate. So one phosphate from UTP and the phosphate from glucose 1-phosphate are removed as a pyrophosphate group to give you UDP glucose. Once UDP glucose is formed, it is now ready to participate in glycogen synthesis. So UDP glucose is the activated form of glucose that participates in glycogen synthesis. Yes? Clear? Okay, so once UDP glucose is formed, now you need another component in order for glycogen synthesis to start. So you have the activated glucose residue ready for glycogen synthesis. But apart from that, you need another component to initiate glycogen synthesis. You either need a glycogen primer. So a glycogen primer can be a pre-existing glycogen molecule present in the cytosol of liver cells as well as muscle cells. Clear? Yes? So in order for glycogen synthesis to start, you need sort of like a foundation on which the glycogen synthesis can occur. You need a foundation on which you can build on. Yes? This foundation can either be a pre-existing glycogen molecule. So a small fragment of a pre-existing glycogen molecule can act as the primer, or there is an enzyme known as glycogen. So this is an enzyme, glycogen. So this glycogen, what it can do is, it can catalyze, it can auto-catalyze the addition of glucose residues to itself. So as you can see here, the first one is glycogen. So glycogen is an enzyme. It can auto-catalyze the addition, or it can catalyze the addition of glucose residues to itself. Is that clear? Yes? Glycogen, it can catalyze the addition of glucose residues to itself. Glycogen is an enzyme. What it can do is, it can catalyze the reaction where new glucose molecules are attached to itself. Yes? So here, as you can see, glycogen, in presence of UDP glucose, have a glucose residue attached to it, isn't it? So the formation or the so once a glucose molecule is attached, again another molecule of UDP glucose is added, where it will join to the glucose residue already present. So the glucose residues in glycogen, they are joined to each other by means of alpha 1,4 glycosidic bond. Yes? I hope you know that. No? You don't? Yes, okay. So here the glucose molecules or glucose residues are attached to each other by means of alpha 1,4 glycosidic bond. And this bond formation is catalyzed by glycogen. Yes? So glycogen will catalyze the formation of alpha 1,4 glycosidic bond so that the glycogen chain elongates or increases or lengthens. Clear? Yes? So once around 7 to 10 glucose residues has been attached to glycogen, so here, so this green, this is glycogen. So once 7 to 10 glucose residues has been attached to it, so you have now a glycogen chain which has, here right now it has 7 residues. Once the 7 residues, you have a chain with 7 residues, another enzyme known as glycogen synthase will take over the formation of alpha 1,4 glycosidic bond. Clear? Yes? Initially, glycogen is responsible for alpha 1,4 glycosidic bond formation. Yes? And at the growing chain, the growing glycogen chain is attached to glycogen, and glycogen will catalyze the formation of alpha 1,4 glycosidic bond so that the chain increases in length. Clear? Once you have a chain that is 7 to 10 residues, glucose residues long, so meaning once you have at least 7 glucose residues in the chain, now glycogen will stop its catalytic activity. Clear? Yes? A new enzyme known as glycogen synthase will now catalyze the formation of the glycosidic bond. Clear? So once you have a glycogen chain that is 7 glucose residues long, a new enzyme known as glycogen synthase will take over the formation of alpha 1,4 glycosidic bond. Yes? Hope that's clear. So alpha 1,4 glycosidic bond, I hope you know what the 1 and 4 represents. Yes? Okay. So in this way, glycogen synthase will now add more and more glucose residues to the non-reducing end of the chain. So to the non-reducing end of the chain, you have new glucose residues carried by UDP glucose. So UDP glucose will bring the glucose residues, and glycogen synthase will catalyze the formation of alpha 1,4 glycosidic bond to the non-reducing end of the chain. Yes? So in this way, new glucose residues are added. So once the glycogen chain it has 14 glucose residues. So here, as you can see, so here you have around 14 glucose residues. Once the chain reaches that length, a branch point is added. So you know that glycogen is a branched polysaccharide, isn't it? it has multiple, numerous branches. So here, once the linear glycogen chain has at least 14 glucose residues, enzyme known as branching enzyme. Branching enzyme, which is also known as 1,4-alpha glucan branching enzyme, or simply as branching enzyme. So this should be easy for you to remember. Branching enzyme is responsible for creating a branch point. Clear? So here you only have two, well, technically three enzymes that you need to know. Glycogen, which serves as a primer. Then you have glycogen synthase, which is responsible for the formation of alpha-1,4 glycosidic bond, so that your glycogen chain increases in length. And the next enzyme is branching enzyme, which is responsible for the formation of a new branch point. And at the branch point, the type of glycosidic bond that is formed is alpha-1,6 glycosidic bond. Clear? In a linear chain, the glucose molecules are attached to each other by means of alpha-1,4 glycosidic bond. But at the branch point, the glucose residue are attached to each other by means of alpha-1,6 glycosidic bond. Clear? So alpha-1,6 glycosidic bond is only formed at the branch point. Clear? In a linear chain, all the glycosidic bond is alpha-1,4. Only at the branch point is a alpha-1,6 glycosidic bond formed. So this branching enzyme, it will transfer a small section of the linear glycogen chain and form a branch point. So here an alpha-1,6 glycosidic bond is formed to form a new branch. So now you have two chains. Yes? Now again, glycogen synthase will add glucose residues to these two chains to elongate the chain. Clear? Yes? And again, when the chains have 14 or more glucose residues, the branching enzyme will form another branch point. Clear? So with the combined action of glycogen synthase and branching enzyme, the cells will synthesize a highly branched glycogen molecule which contains close to 60,000 glucose residues. Clear? So one glycogen molecule will contain almost 60,000 glucose residues. Clear? So the function of glycogen synthase is to join glucose residues in a linear chain by means of alpha-1,4 glycosidic bond. The branching enzyme, it creates new branch by creating an alpha-1,6 glycosidic bond. And the combined action of glycogen synthase and branching enzyme will result in the formation of a highly branched glycogen molecule. Yes?
Why can't glycogen continue one with the type with the one?
7 to 10. So it acts basically as a primer. So once it's reached that substantial amount, now it will stop autocatalyzing because it is unable to, its catalytic effect. It cannot hold on to longer or bigger molecules. So now glycogen synthase will take over that activity. So its major function is just to create a short glycogen chain. And once that short glycogen chain is formed, then glycogen synthase. Because as the chain grows, the enzyme also needs to move along it, isn't it? Yeah, that is why. Okay, so that's about glycogen synthesis. Hope compared to the other metabolic pathways, this is easier. You only have to remember glycogen, which acts as a primer. It does autocatalysis. It forms alpha-1,4 glycosidic bond to attach glucose residues to itself to form a short chain with 7 to 10 glucose residues. Once the chain has 7 to 10 glucose residues, then the elongation step is taken over by glycogen synthase. Glycogen synthase catalyzes the formation of alpha-1,4 glycosidic bond. Once the chain has around 14 glucose residues, a branch point is created. And this branch point is created by the enzyme branching enzyme. At the branch point, you have a alpha 1,6 glycosidic bond. So a small fragment of the main chain is detached and attached to another glucose residue to form the, so here as you can see, a small fragment, the terminal end of the glycogen chain, it is detached and it is transferred to another residue to form a branch point. And at the branch point you have a alpha 1,6 glycosidic bond. So you only find alpha 1,6 glycosidic bond at the branch point. Everywhere else it is the alpha 1,4 glycosidic bond. Clear? Yeah. So glycogen has branches which are located on an average every 10 to 14 glucose residues apart. So here both these chains, they will elongate, and once these chains have at least 10 to 14 residues, glucose residues, then a new branch point will be formed. Meaning you won't have a branch point just next to a already existing branch point. Yeah. So here you have a branch point, meaning you won't have another branch point here. Clear? There should be at least 10 to 14 glucose residues in between each branch points. Yes? Clear? So between each branch points, there will be at least 10 to 14 glucose residues in between. So they are separated by at least 10 to 14 glucose residues. Yes. So all the cells which are capable of glycogen metabolism have glycogen. Glycogen is not found in any other cells, but it is present in hepatic cells, that's the liver cells, as well as skeletal muscle cells, because they are specialized, or one of their function is for glycogen synthesis. So the cells can either use glycogen, or it can, if there is a pre-existing glycogen molecule, then glycogen synthase can add glucose molecules to that glycogen primer. Clear? So you have two options. If there is, so meaning glycogen will be carrying out glycogen synthesis as well. So in a cell, it's not one molecule of glycogen is synthesized at a time. No. You have multiple glycogen molecules which are synthesized simultaneously. Clear? So in your cell, it's not just one glycogen, and you will have numerous copies or numerous glycogen molecules present, and numerous glycogen molecules are being synthesized at the same time. So some of it can be built on glycogen, some can be synthesized on a pre-existing glycogen molecule. Clear? Yes, it can occur as long as you have a glycogen, a pre-existing glycogen molecule. So the prerequisite is either you need glycogen or you need a pre-existing glycogen molecule. Clear? Because you need a foundation on which the polymerization can take place. Sorry? Yeah. No. So branching enzyme, its function is to only create that branch point. I hope that's what your question was. So the function of branching enzyme is to only create this branch point. Once the branch point is formed, then glycogen synthase will elongate this branch chain. Clear? So the function of branching enzyme is to only create this branch point. So what it does is it transfers a fragment of the glycogen chain to form the branch point. Yes, a new branch. So branching enzyme will create this alpha-1,6 glycosidic bond. Once the branch is formed, then glycogen synthase will elongate the new branch. Yes? I hope that answers your question. Yes. So, yes. Sorry? Glycogen. Yes, so as long as there is a pre-existing glycogen molecule, then glycogen synthase can add glucose residues to it. So the prerequisite is either you need a glycogen molecule or you need a small glycogen molecule to serve as a primer on which a new glucose residues can be added. Hope that, yes. Yes. So branching, advantages of a branching is that the number of non-reducing ends is increased. So in this example here, if you didn't have branching, if it was just a linear chain of glucose residues, then that means you only have one non-reducing end, isn't it? So meaning only one glucose residue at a time can be attached to this chain. Yes? But through branching, the number of terminal ends to which glucose residues can be attached is increased. So thereby it increases your rate of glycogen synthesis. Clear? Yes? If it's a linear chain, you only have one terminal, one non-reducing end to which you can only add one glucose residue at a time. But if you have 10 branches, that means you have 10 non-reducing ends to which 10 glucose molecules can be added at a time. So this increases the rate of glycogen synthesis. So branching helps in increasing the rate of glycogen synthesis. Similarly, the breakdown, the rate of glycogen breakdown is also increased, because if it's just a linear chain, then you can only remove one glucose residue at a time, isn't it? But if you have again 10 branches, that means you can remove 10 glucose residues at a time. So this again increases the rate at which glycogen can be broken down. So branching helps in increasing the rate of glycogen synthesis as well as the rate of glycogen degradation. And it also helps with the size of the molecule, because if it's a linear chain, it will occupy more space in the cell, but when it's branched, it becomes more condensed. So it helps, it increases the size of the molecule also. So that's about glycogen. Yes. No, so glycogen is, the glycogen is going to be like the anchor on which the molecule is built on. So after the glycogen has been significantly increased, it can be removed from the molecule, or it can remain attached, doesn't matter. Yeah. You should look at it three-dimensional. But in two-dimension, I can only show you either to the left or the right. But then, you know, it can. I hope that makes sense. So if this is a glycogen molecule, you can have a branch point that way. You can have a branch point this way, this way, this way, this way, any way, isn't it? Because you need to picture the whole molecule in a three-dimension. Yeah? Like here, I won't be able to show that. So it's not just going to be left or right. No, it can, yeah. So that's about glycogen synthesis. Next is glycogen degradation. So here, this is also a small pathway. You only have 3 enzymes involved in glycogen degradation. So glycogenolysis apart from cytosol, it also takes place within the lysosome. So you know lysosomes are organelles in the cell, isn't it? So glycogenolysis. So that's another name for glycogen degradation, glycogenolysis. So please do not confuse. Glycogenesis is glycogen synthesis and glycogenolysis is glycogen degradation. So genesis meaning synthesis, lysis will be degradation. So here glycogenolysis. So here this glycogenolysis takes place within the cytosol as well as within the lysosome of the cells. Now here the regulatory enzyme. So in glycogen synthesis, the regulatory enzyme is glycogen synthase. So by inhibiting glycogen synthase, you inhibit glycogen synthesis. By activating glycogen synthase, you activate glycogen synthesis. So it is the regulatory enzyme. Here in glycogenolysis, the regulatory enzyme is glycogen phosphorylase. So glycogen phosphorylase, it brings about the breakdown of glycogen by means of phosphorylated cleavage of the alpha-1,4 glycosidic bond. So as you can see here, you have a section of the glycogen molecule. Glycogen phosphorylase will break the alpha-1,4 glycosidic bond by introducing a phosphate group. So here at the terminal end, this glucose residue is removed as glucose 1-phosphate. So this terminal glycosidic bond, so again, the glucose molecules are removed from the non-reducing end. So from the non-reducing end, the terminal 1,4 glycosidic bond is removed by a phosphate group, by attaching or by the attack of a phosphate group. So this phosphate group will break the alpha-1,4 glycosidic bond and the terminal glucose residue is removed as glucose 1-phosphate. This reaction is catalyzed by the enzyme glycogen phosphorylase. So glycogen phosphorylase breaks the alpha-1,4 glycosidic bond by introducing a phosphate to remove the terminal glucose as glucose 1-phosphate. So in this way, from the non-reducing end of the glycogen molecule, you have glucose residues being removed as glucose 1-phosphate. Now, so here, as you can see, the purple or the pink hexagonal units, so they represent the glucose units that are removed by the action of glycogen phosphorylase. Once you have only 4 glucose residues on a branch chain, so here you have a branch. So this branch has only 4 glucose residues. So when at the point when the branch has only 4 glucose units, you have an enzyme known as glucan transferase, which will transfer a trisaccharide portion of the branch to the main glycogen branch. So here, glycogen phosphorylase will remove all these pink glucose residues with the help of phosphate, and the glucose residues are removed as glucose 1-phosphate. Once the branch has only 4 glucose residues attached to it, glycogen phosphorylase will now stop its action. Clear? Yes? Here, an enzyme known as glucan transferase will transfer 3 glucose residues from the branch and transfer it to the main branch. Clear? Yes? To the longer branch. Yes? Can you see? So the 3 blue glucose residues are transferred from the branch to the main glycogen chain. Yes? This exposes, or now here at the branch point you only have 1 glucose residue. Yes? So at the branch point you only have now 1 glucose residue. This 1,6 glycosidic bond at the branch point is hydrolyzed by debranching enzyme, also known as amylo alpha-1,6 glucosidase, which is also commonly known as debranching enzyme. So branching enzyme is responsible for the synthesis of 1,6 glycosidic bond, whereas debranching enzyme is responsible for the hydrolysis of 1,6-glycosidic bond. Clear? Yes? So glucan transferase, it transfers 3 glucose residues from the branch to the main glycogen chain, leaving only 1 glucose residue attached at the branch point. This branch point is attacked by debranching enzyme, which hydrolyzes the alpha-1,6-glycosidic bond, and this glucose that is attached at the branch point is released as a free glucose. Now again, glycogen phosphorylase will start removing glucose residues from the non-reducing end by breaking the alpha-1,4-glycosidic bond. So in this way, the combined action of glycogen phosphorylase, glucan transferase and debranching enzyme, glycogen molecule is broken down. Clear? Yes? So glycogen phosphorylase, it breaks the alpha-1,4-glycosidic bond by introducing a phosphate group, and the glucose residue is removed as glucose 1-phosphate. Again, glycogen phosphorylase will continue removing the terminal glucose residues till it reaches a point where there is only 4 glucose residues on the branch. Here, glucan transferase will transfer 3 glucose residues, which is colored in blue. So 3 glucose residues are transferred from the branch to the main or the longer glycogen chain, and this exposes, or this now leaves only 1 glucose residue attached at the branch point. The branch point, which is joined by alpha-1,6-glycosidic bond, is hydrolyzed by the debranching enzyme, and this glucose residue is removed as free glucose. Now again, glycogen phosphorylase will continue removing the terminal glucose residue. So when I say terminal glucose residue, it means from the non-reducing end. So addition of glucose residue occurs at the non-reducing end, and also the release of glucose residues occur at the non-reducing end. So in this way, the combined action of glycogen phosphorylase, glucan transferase and debranching enzyme will result in the breakdown of glycogen to glucose residues, glucose 1-phosphate predominantly. Clear? So once glucose 1-phosphate is formed, then again the same enzyme, phosphoglucomutase, will transfer the phosphate group from C1 to C6 position. Yes? So now you have glucose 6-phosphate. In the muscle cells, the glycogen degradation stops with the formation of glucose 6-phosphate. Clear? In the muscle cell, the glycogenolysis or glycogen breakdown stops with the formation of glucose 6-phosphate. This glucose 6-phosphate now moves into glycolysis so that ATP molecules can be generated, because the function of glycogen in the muscle is to provide glucose for glycolysis, isn't it? So this glucose 6-phosphate enters into glycolysis to generate energy. Whereas in the liver, because the function of the liver glycogen is to maintain blood glucose level, and I already mentioned, phosphorylated glucose cannot move out of the cell, isn't it? It gets trapped. I hope you remember from glycolysis. As soon as glucose enters into the cell, it is phosphorylated so as to trap it, because there is no transporter that can carry glucose 6 or phosphorylated glucose out of the cell. Yes. Hydrolysis.
But how does the combination of both?
It's the hydroxyl group, they attack the bond. Yes. So as to form a glucose. And so that's what happens. The water molecule, the hydroxyl group, it attacks the. So I hope you know those electrophilic attack, nucleophilic attack. Basically, that's what happens. So, anyways, Settle down. Yes. So once glucose 6-phosphate is formed in the muscle cells, the glycogen breakdown stops. The glucose 6-phosphate is channeled into glycolysis so that ATP synthesis or ATP energy formation can occur. Whereas in the liver, the function of liver glycogen is to help maintain the blood glucose level. So you need the phosphate group to be removed from the glucose because phosphorylated glucose is trapped. It cannot move out of the cell. So here in liver, you have an enzyme known as glucose 6-phosphatase. So glucose 6-phosphatase is only present in the liver. It is not present in the muscle. Clear? Yes? So that is why the glycogen degradation stops at glucose 6-phosphate in muscle cells because it lacks or it does not have the enzyme known as glucose 6-phosphatase. Glucose 6-phosphatase is present in the liver as well as in the kidneys, but it is not present in the muscle. So the only two organs or tissues in which glucose 6-phosphatase is present is in the liver and the kidney. So again, phosphatase. So the function must be the removal of phosphate group. So from glucose 6-phosphate, by the action of glucose 6-phosphatase, the phosphate group is removed. And now glucose is free to move out of the cell because again, liver has GLUT2 transporter. I hope you remember. Yes, the unique feature of GLUT2 transporter is that it helps in bidirectional transport, meaning it allows the movement of glucose from outside the cell into the cell as well as from inside the cell to the outside. So because these GLUT2s are present on the liver, once the phosphate group has been removed from glucose 6-phosphate by the enzyme glucose 6-phosphatase, the free glucose is now exported out of the cell and it is now released into circulation where it can be carried to the extrahepatic tissues like the brain and the erythrocytes to be used up as a metabolic fuel. Yes? So lysosomal degradation is carried out. So only a small amount, that is 1 to 3% of glycogen degradation takes place within the lysosome, and the enzyme which is responsible for the breaking of the alpha-1,4 bonds are alpha-1,4 glucosidase. Alpha-1,4 glucosidase, lysosomal alpha-1,4 glucosidase. So as you can see here, only a small amount of glycogen degradation takes place because in the lysosome it lacks the enzyme required to break the alpha-1,6 glycosidic bond. So only those residues at the terminal ends will be removed. But once the branch points are reached, it will be released back into the cytosol so that the debranching enzyme can break the alpha-1,6 glycosidic bond. So further glycogen degradation takes place within the cytosol. Only the external glycogen residues are removed by the lysosomal 1,4 glucosidase enzyme. The remaining glycogen or the complete glycogen degradation then occurs within the cytosol because the debranching enzyme is present within the cytosol. Clear? Okay, so that's with the glycogen synthesis and glycogen degradation or glycogenesis and glycogenolysis. Just recapping again, glycogen synthesis, the regulatory enzyme is glycogen synthase and then which catalyzes the formation of alpha-1,4 glycosidic bond, and the branch points are formed by branching enzyme which will form alpha-1,6 glycosidic bond. In glycogenolysis or glycogen degradation, the regulatory enzyme is glycogen phosphorylase. It breaks the alpha-1,4 glycosidic bond by introducing a phosphate group. In this way, glycogen phosphorylase will remove the glucose residues from the terminal or the non-reducing ends of the glycogen molecule till there's only about 4 glucose residues on a branch. The 3 of the glucose residues are transferred from the branch to the main glycogen chain by glucan transferase. So all that remains at the branch point is 1 glucose residue, which is the alpha-1,6 glycosidic bond is broken by the debranching enzyme releasing a free glucose molecule. So these are the enzymes involved in glycogenesis and glycogenolysis. So as with any kind of metabolic pathway, it needs to be regulated because there are certain conditions when glycogenesis is favored and certain conditions where glycogenolysis is favored. So this depends on the metabolic needs of the cell or the metabolic needs of the body. If there is an abundance of glucose, then it is favorable for glycogenesis to take place, isn't it? Yes? When there's an abundance, an excess of glucose, what do you do with this excess glucose? You store it. Yes? So the condition is now favorable for glycogen synthesis. So you need all the enzymes or the regulatory enzyme of glycogen synthesis to be activated. Whereas in the case of fasting, during fasting, there is no glucose, excess glucose available. So the conditions are unfavorable for glycogen synthesis, isn't it? Yes? When you're fasting, your blood glucose levels are low. Is it a favorable condition for glycogen synthesis? So what happens? You need to inhibit the enzyme glycogen synthase. So glycogen synthesis does not occur, isn't it? Then during fasting, what do you need? You need more glucose because your blood glucose levels are low. So which pathway should be activated? Glycogenolysis, yes, the breakdown of glycogen so that glucose can be released into the blood to increase the blood glucose level. So in these conditions, in these metabolic conditions, the regulatory enzymes are either inhibited or activated, and they can be inhibited or activated via hormonal response or by means of allosteric effectors. So hormonal response brings about the activation or inhibition of the enzyme by covalent modification. So here the covalent modification is the addition or attachment of a phosphate group. Clear? So here, by the addition of a phosphate group to a cell, you either activate it or inhibit it. So the hormonal response is the three hormones which play a part in the regulation of glycogen metabolism are glucagon and epinephrine, which are released when your blood glucose levels are low, and insulin, which is released when your blood glucose levels. So glucagon and epinephrine, it is released when your blood glucose levels are low. So which pathway would it favor? Huh? Yes, glycogenolysis. So glucagon and epinephrine, it is released when your blood glucose levels are low. So it will favor the activation of glycogenolysis. So it will favor glycogenesis, isn't it? It will favor the synthesis of glucose because insulin's presence in the circulation means there is an abundance of glucose, and this is favorable for glycogen synthesis. So the regulatory enzymes that are for glycogen synthesis is glycogen synthase, and for glycogenolysis is glycogen phosphorylase. So they are regulated by reversible phosphorylation and dephosphorylation by means of certain enzymes. Now cyclic AMP. So here the phosphorylation of the enzymes is brought about by cyclic AMP, which is formed from ATP in presence of the enzyme adenyl cyclase. So this adenyl cyclase will convert ATP in the cells to cyclic AMP, and all the responses of the hormones such as epinephrine and glucagon are brought about by cyclic AMP. So glucagon and epinephrine will bring about the formation of cyclic AMP, which will now carry out the regulatory action. Increased cyclic AMP leads to phosphorylation of proteins, resulting either in their activation or inhibition. Glycogen synthase, it exists in two forms. In the phosphorylated form, it is inactive. Glycogen synthase in the phosphorylated form is inactive. In the dephosphorylated form, it is active. Clear? So phosphorylated form is meaning it has a phosphate group attached to it. In that form, it is inactive. Whereas in the dephosphorylated form, that is when the phosphate group is removed, it becomes active. In the case of glycogen phosphorylase, the opposite is true. When it is phosphorylated, it is active. When it is dephosphorylated, it is inactive. Clear? So what I would suggest is memorize one of those. So for glycogen synthase, phosphorylated form inactive, dephosphorylated form active. In glycogen phosphorylase, it will be the opposite. Yes? So in that way, you just memorize it. So glycogen phosphorylase is active in its phosphorylated form and inactive in its dephosphorylated form. Insulin brings about dephosphorylation of proteins by activating an enzyme known as protein phosphatase 1. So cyclic AMP will bring about phosphorylation, whereas insulin via protein phosphatase 1 enzyme will bring about dephosphorylation. Now that is about the covalent modification or hormonal regulation of enzyme. Now allosterically, glycogen phosphorylase. So glycogen phosphorylase, it is an allosteric enzyme. So it can be activated or inhibited by means of allosteric effectors. So here, glycogen phosphorylase is allosterically inhibited by glucose 6-phosphate as well as ATP. So when the levels of glucose 6-phosphate and ATP are high, it can allosterically inhibit glycogen phosphorylase. Clear? Glycogen phosphorylase, it is an allosteric enzyme, so it can be inhibited in presence of high levels of glucose 6-phosphate and ATP. So glucose 6-phosphate and ATP, it can bind to the allosteric site of glycogen phosphorylase and inhibit the enzyme action. So here, this is the going to explain in detail the hormonal regulation of glycogen synthesis and glycogen degradation. So as I already mentioned, glucagon. So glucagon has receptors only on the liver, and the muscle cells epinephrine. So epinephrine has receptors both in the muscle as well as the liver, whereas glucagon has only receptors on the liver. So when glucagon and epinephrine are released in the circulation, they bind to their receptors which are present on the plasma membrane. Once the hormone receptor complex is formed, it will activate the enzyme adenyl cyclase, which will convert ATP to cyclic AMP. Cyclic AMP will activate an enzyme known as cyclic AMP dependent protein kinase. So here, protein kinase. What will the function be? Phosphorylation, yes. So cyclic AMP, when the levels of cyclic AMP increases, it will activate an enzyme known as cyclic AMP dependent protein kinase, whose function is to phosphorylate. So here, protein kinase, it will phosphorylate glycogen synthase. So what happens when glycogen synthase is phosphorylated? It becomes inactive, isn't it? So cyclic AMP dependent protein kinase will phosphorylate. So as you can see here, the glycogen synthase, this is the activated form. It gets phosphorylated by cyclic AMP dependent protein kinase. So here's a phosphate group attached to it. Once a phosphate group is attached to it, it becomes inactive. And when glycogen synthase becomes inactive, it means glycogen synthesis is inhibited. Yes? Because if glycogen synthase is, then the elongation of the glycogen molecule does not take place, isn't it? So glycogen synthesis becomes inhibited. So this is when your blood glucose levels are low and when glucagon and epinephrine are secreted. Now what happens when your blood glucose levels are high? Insulin is secreted. So insulin will bring about the activation of the enzyme protein phosphatase 1. Enzyme is phosphatase. So what is the function? Dephosphorylation. So here protein phosphatase 1 will remove the phosphate group. So as you can see here, it will remove the phosphate group, and once glycogen synthase is dephosphorylated, it becomes active and glycogen synthesis can occur. Clear? Yes? So insulin will activate glycogen synthase by dephosphorylating it with the help of the enzyme protein phosphatase. Once it is activated, it will carry out glycogen synthesis. Is this clear? Okay. So next is the regulation of glycogenolysis. Again, it's the same as glycogenesis when glucagon and epinephrine bind to their respective receptors on the plasma membrane. The hormone receptor complex will activate adenyl cyclase, which will convert ATP to cyclic AMP. As the levels of cyclic AMP increases, it activates the enzyme cyclic AMP dependent protein kinase. Now here there is an extra enzyme, because in the case of glycogenesis it was pretty straightforward. Cyclic AMP dependent protein kinase directly phosphorylates glycogen synthase, isn't it? Here there is another enzyme, glycogen phosphorylase kinase. So glycogen phosphorylase is the name of the substrate. Kinase is the activity. And kinase, the activity is phosphorylation, yes? Yes? So glycogen phosphorylase kinase is phosphorylated by cyclic AMP to its activated form. So in the dephosphorylated form, this enzyme, glycogen phosphorylase kinase, is inactive. It is phosphorylated by protein kinase A to give you glycogen phosphorylase kinase. So this is the activated form. Once this is activated, again it will phosphorylate glycogen phosphorylase to its activated form. Once activated, it will bring about glycogenolysis. Clear? So cyclic AMP dependent protein kinase will phosphorylate and activate glycogen phosphorylase kinase. Yes? Yes? Cyclic AMP dependent protein kinase will phosphorylate glycogen phosphorylase kinase. Yes? Okay, so here cyclic AMP dependent protein kinase, so that is pKa, is cyclic AMP dependent protein kinase A. Once activated, it will phosphorylate glycogen phosphorylase kinase. So this is the name of the substrate. Clear? Yes? This is the, so enzyme classification, you know that the name either is the substrate name, and then the second is the type of enzyme, isn't it? So here the name of this enzyme is glycogen phosphorylase kinase. So the substrate is glycogen phosphorylase. Yes? So meaning it acts on glycogen phosphorylase. Kinase is the type of reaction, or is the enzyme activity. Kinase meaning it phosphorylates. It's a type of enzyme that phosphorylates. Clear? Yes? So here, this is also a kinase. So what does it do? It will, so here glycogen phosphorylase kinase, this is a dephosphorylated form, meaning there's no phosphate attached to it. That is inactive. Yes? In presence of protein kinase A, a phosphate group a phosphate group is added to glycogen phosphorylase kinase. So now you have glycogen kinase to which a phosphate group is attached. To which a phosphate group is attached, and this is now active. Clear? Protein kinase, its function is to phosphorylate. So it phosphorylates glycogen phosphorylase kinase to its active form. Clear? Yes? Once this enzyme is active, again, so what is its function? What is the substrate? It's going to act on? Yes, so the function or the substrate of this enzyme is glycogen phosphorylase. What is the action going to be? Phosphorylation. So again, this enzyme will convert glycogen phosphorylase. So this is the dephosphorylated form. So it is inactive. This enzyme will add a phosphate group. So the phosphate group is provided by ATP. So ATP will donate one of its phosphate groups to give you glycogen phosphorylase, which is phosphorylated, and this is the active form. Yes? So once activated, glycogen phosphorylase, once it's activated, it will now carry out glycogenolysis. Clear? Yes? Again, glucagon and epinephrine will bring about the activation of glycogen phosphorylase, meaning it favors glycogen breakdown. Clear? Whereas in the case of insulin, which is secreted during high or when your blood glucose levels are high, your blood glucose levels are high, you don't need now more glucose to be released by the liver, is there? So insulin is secreted when your blood glucose levels are high. Do you need more glucose to be released by liver? No, because already the blood glucose level is high. Why do you need to pump more glucose into the circulation? So you need glycogenolysis to stop, isn't it? Yes? So insulin again activates protein phosphatase. The action of protein phosphatase is to dephosphorylate glycogen phosphorylase kinase. So here again, protein phosphatase 1, it acts on glycogen phosphorylase kinase and not on glycogen phosphorylase. Clear? Yes? So insulin, through the action of protein phosphatase 1, will dephosphorylate glycogen phosphorylase kinase to its inactive form. Once it's inactivated, it will stop further activation of glycogen phosphorylase and your glycogenolysis is inhibited. Yes? It stops. So these are the hormonal response. Now there are non-hormonal response or so meaning even in the absence of the hormone, there are certain effectors that can activate glycogenolysis. So the first is the role of calcium in muscles. So here, this is a mechanism that occurs within the muscle. Have you started muscle contraction? Yes? So you know the importance of calcium ions in muscle contraction, isn't it? So during muscle contraction, calcium ions will be released. So what this calcium ion does is, it binds to glycogen phosphorylase kinase and activates it. So here, calcium, it binds to glycogen phosphorylase kinase and activates it, bringing about glycogenolysis. So here it is sort of an allosteric effector of glycogen phosphorylase kinase. Clear? Yes? So here it is an allosteric activator. Calcium is an allosteric activator of glycogen phosphorylase
kinase. When the concentration of calcium in the cells increase, meaning there is a need for ATP molecule, or there's a need for energy, isn't it, to carry out muscle contraction? Yes? So you need glycogen to be broken down to glucose so that it can enter into glycolysis to generate more ATP molecules. So calcium will bind, it will allosterically bind to glycogen phosphorylase kinase and activate it, which in turn will activate glycogen phosphorylase and bring about glycogenolysis. Clear? Yes? So here, in this case, there is no phosphorylation taking place. Yes? It activates allosterically, meaning it binds to the allosteric site of glycogen phosphorylase kinase and activates it. Yes? Similarly, AMP, it allosterically binds to glycogen phosphorylase and activates it. Yes, we already know that glycogen phosphorylase is an allosteric enzyme because I had mentioned glucose 6-phosphate and ATP allosterically inhibits glycogen phosphorylase. Remember from 2 slides ago? Yes, glucose 6-phosphate and ATP, it will allosterically inhibit glycogen phosphorylase. So we know that it's an allosteric enzyme. Yes? So here AMP will allosterically activate by binding to glycogen phosphorylase. So here again there is no phosphorylation taking place. AMP binds to the allosteric site and activates the enzyme. And once the enzyme is activated, glycogenolysis will take place. Yes? So the role of AMP and calcium, it occurs within the muscle because when you are performing muscular activity, your blood glucose level need not necessarily be low. So glucagon won't be released. Yes? But epinephrine will be because fight or flight. Yes? So epinephrine can bind to the its receptor and carry out the hormonal regulation. But in the case of, if you're just performing some minor muscular activity, you need a constant supply of ATP. So in this case, instead of waiting for epinephrine to be released, to bind to its receptor and so on, calcium, which is released into the cytoplasm, will activate glycogen phosphorylase kinase, bringing about breakdown of glycogen so that glucose 6-phosphate is formed, which can be diverted into glycolysis to produce more ATP molecule. Similarly, when your energy levels in the cell, in the muscle is low, so meaning when you say your energy levels low, you mean high levels of AMP. When a cell's energy levels are high, that means there's high levels of ATP. Clear? Yes? When the energy levels of the cell are high, the levels of ATP will be high. Clear? Whereas when you say the energy levels of a cell is low, then it will have more of AMP. Clear? So here the high levels of AMP meaning there's not enough ATP in order to perform your muscular activity. So AMP will activate glycogen phosphorylase to carry out glycogenolysis so that more glucose can be diverted into glycolysis to produce ATP molecules. Clear? So these are the hormonal regulation of glycogenesis and glycogenolysis, as well as the non-hormonal regulation via calcium ions and AMP, which takes place within the muscle. So we move on to the last part, which is the clinical significance of glycogen metabolism. So here, any defect in the enzymes responsible for glycogen metabolism can bring about certain disorders. So they are collectively known as glycogen storage disease. So there are a series of inborn errors. So these are genetic disorders. They are inborn errors of metabolism, and they collectively known as glycogen storage disease, and they might be as a result of the deficiency or defect in the enzymes of glycogen synthesis or glycogen degradation. Muscle glycogen phosphorylase, it is genetically different from liver glycogen liver glycogen phosphorylase. So muscle glycogen phosphorylase and liver glycogen phosphorylase, even though they catalyze the same kind of reaction, they are genetically different, meaning their composition is different. Clear? So these two proteins, they have different amino acid composition. That's what it means. They are different. Even though they catalyze the same type of reaction, they are different. They are coded by different genes. That's why they're called genetically different, because they're coded by different genes. And it will become more clear when you start with protein synthesis, the genetic part, when I say they're coded by different genes. So just know that they are genetically different. They are not identical enzymes. They are two different types of enzyme which catalyzes the same type of reaction. So some of the degradation of glycogen also occurs within the lysosome, which we already saw. So a defect in the lysosomal enzyme can also bring about glycogen storage disease. So here, when the lysosomal enzyme is defective, the glycogen particle, they get surrounded by membranes and they fuse with the lysosomal membrane and they form these granules in the cells. Lysosomal glucosidase, it hydrolyzes the glycogen to glucose. Already mentioned that. So we're going to discuss 6 types of glycogen storage disease. There are more glycogen storage disease. There are around 11, 12 types of glycogen storage disease. I'm just going to mention the common and important ones here. So the type 1 is known as von Gerg's disease. Type 1 is known as von Gerg's disease. So please, you need to know von Gerg's disease, that it's type 1. Yeah? You need to know the type and the name of the disease. So von Gerg's disease is the type 1 glycogen storage disease, and here the enzyme defective is glucose 6-phosphatase. So glucose 6-phosphatase, so meaning the glycogen metabolism in the liver is affected, isn't it? Yes? Because glucose 6-phosphatase is only present in the liver, it is not present in the muscle. So here the glycogen metabolism in the liver is affected. In type 1 glycogen storage disease, the glycogen metabolism is affected in the liver. Absence of glucose 6-phosphatase in the liver causes hypoglycemia, meaning the glucose 6-phosphate remains trapped within the hepatocytes, isn't it? Yes? Because glucose 6-phosphatase is not available to convert it to free glucose, which can then move out of the cell. Instead, because of the absence of this enzyme, the glucose 6-phosphatase remains trapped within the hepatocytes, and because there is no glucose being released into circulation, there is hypoglycemia, which means low blood glucose. Yes? I hope you remember from when I said hyperglycemia, meaning high or abnormally high levels of blood glucose, whereas hypoglycemia means low blood glucose levels. So because glucose 6-phosphatase is absent, there is hypoglycemia, because glucose cannot be formed from glucose 6-phosphatase. The hypoglycemia in type 1 glycogen storage disease is said to be severe. This is because glucose 6-phosphatase, apart from being part of the glycogen degradation pathway, it is also involved in glyconeogenesis. So glyconeogenesis is a pathway in which glucose is synthesized from non-carbohydrate precursors like pyruvate, lactate, different intermediates of citric acid cycle. So meaning your body is capable of synthesizing glucose from precursors such as pyruvate, lactate, glycerol. Clear? And in order for glucose to be synthesized from these precursors, you again require glucose 6-phosphatase. So in other words, if gluconeogenesis occurs and there's something wrong with glycogen degradation in liver, the hypoglycemia is not very severe because you have glucose being synthesized through gluconeogenesis. Clear? So, glycogenolysis and gluconeogenesis. The function, so let me just put this as liver. So liver glycogenolysis and gluconeogenesis, their function or their end product is free glucose. Yes? The function of liver glycogenolysis and gluconeogenesis is to produce or to synthesize glucose. Yes? Now both these pathways, they require glucose 6-phosphatase. Yes? Both these pathways require glucose 6-phosphatase. So when there is a defect in glucose 6-phosphatase, it means there is no synthesis of glucose occurring in the body. Yes? And this can lead to severe hypoglycemia. Yes? But if some other enzyme in glycogenolysis, say debranching enzyme, is defective, the hypoglycemia is not very severe because you still have glucose 6-phosphatase which can carry out some of the glucose synthesis. Yes? I hope that's clear. Okay. So in type 1 glycogen storage disease, the did it switch off? Yeah, I think you have your charger. So type 1. So in type 1 glycogen storage disease, you have, yes, severe hypoglycemia. That's because glucose 6-phosphatase is affected, which results in severe hypoglycemia, meaning gluconeogenesis as well as glycogen metabolism or glycogenolysis in the liver does not take place. So there is no glucose being released into circulation. So that's type 1 glycogen storage disease. Type 2 is known as the lysosomal. In type 2 glycogen storage disease, which is known as Pompe's disease, the enzyme that is defective is lysosomal glucosidase. So the enzyme metabolism or the lysosomal glycogenolysis is affected, and because of this, glycogen accumulates in the lysosomes, and when glycogen starts accumulating in the lysosome, they form a membrane-bound mass, which now starts interfering with the function of the hepatocytes. So they also affect, apart from hepatocytes, they also affect the heart, skeletal muscle, liver and nervous tissues, because I mentioned small amounts of glycogen metabolism occurs in the brain, the kidney, the liver, the heart muscles and so on. So this lysosomal glucosidase, it also affects the glycogen metabolism in these tissues, and they can interfere with the function of the cells in these tissues. Next is type 3 glycogen storage disease, which is known as Cory's disease. In Cory's disease, the enzyme defective is the branching enzyme. Okay, so there's no problem. Okay, so in type 3 glycogen storage disease, which is known as Cory's disease, the enzyme which is defective is the debranching enzyme. So here again, debranching enzyme being defective means your glycogenolysis is affected, isn't it? Meaning your branch points cannot be it cannot be hydrolyzed or it cannot be broken. So here the hypoglycemia is not so intense because you still have some, the terminal glucose residues can be removed, isn't it? Which is the branch which when there is 4 glucose residues, that's when your glycogenolysis will stop. Yes? Yes or no? Yes. Like debranching enzyme, its function is to remove. Okay, so the debranching enzyme, its function is to remove or to hydrolyze the alpha-1,6 glycosidic bond. Now, because the debranching enzyme is defective, the branch points will not be removed. So the terminal ends or the external glucose residues can be still removed by the function by the action of glycogen phosphorylase. So the amount of or the hypoglycemia is not very severe because you still have glucose which is released into circulation. Yes? Yes? And in the case of muscles, still it still has some glycogen or some glucose available to it when the terminal glucose residues are removed. What is this announcement that you want to do?
Okay, there's a discussion we're having as class reps, but to explain, it's about the biochemistry class on Monday.
Then it's fine. Okay, let me just. All right. So type 3 Cory's disease, as I was mentioning, it affects, or the enzyme defective here, defect is in debranching enzyme. Debranching enzyme is present in both the liver and the muscle cells. So it affects both the glycogen metabolism or glycogenolysis in liver as well as the muscle cells. So here, glycogen is abnormal and it accumulates, affecting the function of the tissues. Because as I mentioned, the glycogen is not completely broken down, isn't it? The branches cannot be removed, and as a result, you still have a glycogen molecule which is accumulated in the cells, and as they accumulate in the cells, they interfere with the cellular function in the liver as well as in the skeletal muscles. So only the outermost branches of the glycogen are hydrolyzed or are broken down to release glucose. So a small fraction of this abnormal glycogen is functionally active because it still you can still remove glucose from the terminal ends of the chain. Next is type 5 glycogen storage disease, which is known as McArdle disease. Here, the enzyme which is defective is muscle glycogen phosphorylase. So, meaning the glycogenolysis in the muscle is affected because muscle glycogen phosphorylase is defective. Now, the individuals with this type of disorder or disease does not exhibit any type of symptom other than unusual fatigue and muscle cramps during exercise because it is not a serious disease. As you know, the function of muscle glycogen is to provide glucose for glycolysis in the muscle, isn't it? During muscular activity. So individuals are usually without any symptom except when they start performing certain physical activity, then they get muscle cramps and your fatigue. That's because not enough glucose is being available, not enough glucose is available to the muscle in order to carry out glycolysis and generate ATP molecule. So they just suffer from fatigue. They just suffer from fatigue and muscular cramps. So it's not very serious condition. And as long as, and if they're not performing any strenuous activity, then they are without any symptom. The last glycogen storage disease is known as Hers disease or type 6 glycogen storage disease. And here the enzyme which is defective is liver glycogen phosphorylase. So McArdle disease, the enzyme defective is muscle glycogen phosphorylase. In Hers disease, the enzyme defective is liver glycogen phosphorylase. So here, because the liver glycogen phosphorylase is defective, there is hypoglycemia. The individual suffers from hypoglycemia because again, the function of liver glycogen is to provide glucose, free glucose into circulation and maintain the blood glucose levels, isn't it? If there is a defect in the liver glycogen phosphorylase, meaning glucose is not being released from the into circulation, yes or no? Yes. So if there's no glucose being released into circulation, that means your blood glucose levels are not maintained and the individual will suffer from hypoglycemia. Now the hypoglycemia in Hers disease is not as severe as in von Gog's disease. Clear? Because in Hers disease, even though glucose is not released as a result of glycogenolysis, there is still glucose synthesis as a result of gluconeogenesis. Clear? Yes? So the hypoglycemia in Hers disease is not as severe as that seen in von Gog's disease, because here gluconeogenesis can be still carried out to produce glucose which is released into circulation. So the hypoglycemia here is usually mild because again the blood glucose levels can be maintained as a result of gluconeogenesis. But accumulation of the liver can lead to enlargement, accumulation of glycogen in the liver can lead to enlargement of the liver. So meaning it will affect with the liver function. So initially it's fine, but as the disease progresses, it can affect with the hepatic function or the liver function because there is more and more glycogen being accumulated in the liver. Clear? So when your blood glucose levels are high, you have more glycogen which is synthesized, but it is not being broken down. So as more and more glycogen starts to accumulate in the hepatocytes, it will interfere with the function. There is going to be liver enlargement because of the accumulated glycogen, which will now interfere with the liver function. So these are the different types of the important and commonly found glycogen storage disease. Yes. So the most important thing that you need to understand and remember is that as long as the glycogenolysis affects liver the liver, there is going to be hypoglycemia because the function of the liver glycogen is to maintain blood glucose level. So if your glycogenolysis in the liver is affected, then there is going to be some form of hypoglycemia here. So in Cory's disease, where there is a defect in the debranching, and in Hurst's disease, the hypoglycemia is going to be mild because you still have gluconeogenesis to generate glucose to be released into circulation. But in the case of von Gert's disease, where there is deficiency of glucose 6-phosphatase, the hypoglycemia is severe because here both glycogenolysis as well as gluconeogenesis is not occurring. So the hypoglycemia is severe in that case. Yes? And also remember that muscle glycogen phosphorylase is different from liver glycogen phosphorylase. They are genetically different. They are two different types of, they are structurally different enzymes, but they catalyze the same type of reaction. So you can say they are like isozymes. I hope you know that term, isoenzymes. Yes? Which are enzymes which are different, genetically different, but they catalyze the same reaction, isn't it? Yes, so those are just minor they're very rare glycogen storage disease. That's something that you guys can read up on your own. I just mentioned the main type, the common and the main type of glycogen storage disease. As I mentioned, there are I think 13 or 15 types of glycogen storage, but these are very rare. So yeah, I've just mentioned the most commonly occurring glycogen storage disease. Okay, so with that, we've come to an end in carbohydrate metabolism. Starting next week, you will be starting with lipid metabolism, and Dr. Omar will be taking those lectures. All right.